is still working on this and is still working on this project. I want to thank Kenza Araba, who I've known for a very long time. She's my dear neighbor and now is working in my lab. She's gonna hear next week if she is going into veterinary school. I wish her all the best. She's getting her COVID-19 vaccine here with me the same day. So that was pretty fun and an exciting moment. I wanna thank Kendall Schaefer, our cell guru. She's growing all the cells for us that get infected by COVID-19 viruses. And Danielle Williams that has joined the lab very recently. She's a graduate student and she wants to study virology. Our collaborators here at the bottom, Dr. Ralph Barrick, who is an expert here at UNC, is a coronavirus expert, and his student, Caitlin Edwards, that infects cells for us. And Takanori Asakura and Lisa Morton are doing virus gene expression profiling for us. And Victoria Madden is helping us with electron microscopy, which we have used to publish in the New England Journal. So on that, to break the ice, I thought I would give you a little fun facts about me. So when I came to the US in 2000, during my PhD program, I planned to stay only for 8 months. And here I am still 21 years later. So that's what happens to a lot of us, I suspect, on the call. And before I came to the US, before I decided to become a scientist, I considered becoming a scuba diving instructor. So instead of being cool with sunglasses, I became geeky with some glasses looking at microscopes all the time. So another fun fact is I love studying animal behavior. And when I stopped being a personal chauffeur for my kids, which I do all the time, I'm a soccer mom, I admit that. I want to help train another species with behavioral issues, which are dogs. I love dogs. And so another fun fact for me is my worst fear is ignorance. But yet I'm going to talk to you about viruses, and I am neither a virologist or an epidemiologist. So let's see how that goes. So I will give you this slide as a talk overview. We'll talk about the emergence of infectious diseases through the centuries. We'll talk about how COVID-19 came from a food market in Wuhan, China, to a global pandemic. We will talk about the situation right now in the world, the US and in North Carolina more particularly. We will talk about coronaviruses in general and the common colds, and why and how is SARS-CoV-2, the virus that gives us COVID-19, is different from those other coronaviruses. And also, how do we get back to the life that we knew before COVID-19? I think that's what people want the most. I'm going to say it right now, it's vaccines and masks, a little bit longer, but we're seeing the light at the end of the tunnel. So infectious diseases throughout the centuries. So as humans started to move around and travel, they have a little friends with them. They have infectious diseases following them everywhere they go. And this started very early in the centuries. And we had problems with plagues very often in the earlier centuries, which is bacterial caused disease. Yersinia pestis is the bacteria that is responsible for the plagues. And we had a lot of death with the bubonic plague, the La Peste Noire. We had about 200 million dead in the 14th century. So this one was a big killer for sure. But then we have the discovery of antibiotics with Alexander Fleming in 1920 that takes care of those bacterial diseases. The problem that is arising is very commonly nowadays, more commonly, we have viruses outbreaks. So we have the Spanish flu in 1918, and in 1981, we had HIV that started rising. And very recently, actually, we started having in 2002, the SARS, the first SARS infection, which is standing for severe acute respiratory syndrome. And we had a few years later, 10 years back, the Middle Eastern respiratory syndrome, or MERS. So we got infected recently with a new coronavirus called SARS-CoV-2, and that shares about 70% of its genetic sequence with SARS-1 that arose in 2002. So that's the evolution of infectious diseases throughout the centuries. So here I invite you, if you are looking for resources to get numbers on a daily basis, I invite you to look at the Johns Hopkins dashboard, which is very informative, and you find a lot of information. So we know that SARS-CoV-2 started in a food market in Wuhan here, and you can see now the extent that this virus took. It traveled around the world, and it has infected all of the continents and all of the countries. So it's quite amazing in 1 year how the extent of this virus. So right now, I think this is as of yesterday, but I think as of today, we have 112 million global cases in the world. Unfortunately, we have 2.5 million people that died of COVID-19 in 1 year, which is quite a bit. You can go on this dashboard and you can click on the country you're interested in, for instance, the US here, France or Italy, for instance, and you can have the numbers for the death or recovered patients. So we know that we passed the sad bar of 500,000 deaths in the US as of Monday. One thing that I like about this website also is that it shows you the trends, and here you can see the daily death, and we know that we are on the slope down right now, which is really great news. So here, another thing that I like to look at is the trend for the outbreak evolution in the most affected countries. You can see the United States, we didn't do so well, I have to admit. We had several peaks that you can see here. And very recently, we have this dramatic drop down. So we know this is not the effect of vaccine yet. It will come, but maybe a change in leadership has made people realize that wearing masks is actually very important to cope with this infective virus. So you can see also in France, which is very interesting, it's that pink line that you see here. You see those peaks and then they go down, and those are correlating with the confinement that the French government has imposed upon the population. Same for the UK here in blue, or Italy. I suspect it's like this red one here, and you can see very early on those peaks that went down, stayed down for a long time, then they go back up and they are a little more severe with their rules now, and it's going back down again. So we are doing better in the world as we are learning about this virus. So you can go into all the states that you want and you can see the trends. You can see here those trends. So we know we have a lot of people from North and South Carolina here. So you can see that everybody is pretty much going down at this point. I know I have a few people from Texas and a few people from Michigan. So you guys are doing good as well. But in general, the US is doing better at this point. So what is the situation in North Carolina? You can also ask about that, and you have numbers now, not only curves, but you have numbers. So we know we have about 850,000 people that were confirmed cases, about 11,000 dead from COVID-19. The positivity rate of testing is still a little high in North Carolina. We want this number to be below 5 to say that the transmissibility is under control, or the infection is under control. So it's at 8%. I want it to go down a little more. So in North Carolina, we have a total population of about 11 million people. We have administered 2 million doses. So that's first dose. And then we have fully vaccinated with 2 doses. We have 750,000 people. So we know we have a little bit of immunity after the first dose, at least after 2 weeks. So about 20% of the population now will have built immunity probably in North Carolina thanks to the vaccines. You can also look at the hospitalizations, which is great. We want this number to be down. We want to have availability for people to come and get the treatments they need. We have the ICU beds, and we can see the proportion of the beds that are taken by COVID-19 patients. So this is wonderful resources. I invite you to look on a regular basis at your county, at your state, look how you're doing, and just know how the virus is propagating in your surroundings. So we have demographics also. We can see those round circles are the new daily cases per 1,000 people. And I am here in Orange County, and we are doing fairly well here. People wear their mask, they wash their hands, they keep social distancing. So we are doing fairly well in Orange County. Robeson County right now is the bigger threat. It's the biggest threat. We have a lot more positivity rate right there. We can also see the breakdown in racial disparities. So we know that Latinos represent only 9% of the population. However, they represent 20%, 21% of the positive cases. They are doing fairly well health-wise. African Americans, unfortunately, don't do as well in their death rate as other populations. So we have to look into that and try to solve some of these problems still. So COVID-19 has had unparalleled human and economic suffering. Worldwide, like I said, we have about 110 million cases, 2.5 million deaths. That's one too many. We don't want anybody to die anymore of COVID-19. And so we have a percent death rate above 2%. Right now, the daily death rate is about 7,000. It used to be 18,000. So we are dropping that number, which is great news. The US has a total of 28 million cases, 500,000 deaths, and the death rate is below the average, below 2%. So we are still dealing with 2,000 deaths per day. We know that at this point, we have more than 10% of the world's population that has been infected in less than a year, or in about a year. We have a global economic loss of about $8 to $22 trillion in 2020 alone. So we see what the numbers are for 2021. We know people are still struggling with that. The unemployment rate soars at 30 million a few months ago, maybe a couple of months ago, and is now getting better at around 10 million in the US. But I want to emphasize the burnout from our healthcare workers, which are our heroes. If there are anybody on the call anybody that is taking care of COVID-19 patients, thank you so much for your patience. I know it's a very hard work right now, but we are very, very thankful to your effort and your work. So here is another view maybe of something is missing, yes, of the infectious diseases and how they have killed people. We know that the bubonic plague has killed the most, the La Peste Noire, bacteria killed about 200 million people. But COVID-19, being only a year old, has made the top 10 of the most killers infectious disease and is now competing with the 17th century Great Plague. So we are getting vaccinated, but hopefully we can stop that progression very rapidly. Let's not forget that HIV has killed a lot more people, but it's been 40 years in the making, okay? So what have we learned about SARS-CoV-2? And I'm gonna now switch a little bit into the basic research and things that we do in the lab. So first of all, I wanna mention a little bit the symptoms of COVID-19. So 90% of the people will develop a fever. A third of the population will have sputum production, while the 2/3 will have just a dry cough. the common fatigue, sore throat, headaches, chills, we know that these are symptoms as well. We have a small population, small proportion of the population that will develop GI symptoms. But what is very particular about COVID-19 is the loss of taste and smell. So if that happens, you can be asymptomatic, but that's one of the symptoms, but you don't have to have any of the other symptoms to start wondering if you have COVID-19. So here is just showing you graphs of the proportion of infections, symptoms, and hospitalization by age group. So we are starting from the younger individuals in blue to the elderly in red here. And you can see this is a larger group here in green. This is the biggest group, but you can see they are driving pretty much the transmission of SARS-CoV-2. We know that younger people have a tendency to go out and mingle with other people, and that drives the rates up. And the problem is so between infection and symptoms, we have a good balance, but where it's imbalance, it's the hospitalization. People that are getting older are getting more often hospitalized than younger patients. So another resource is the CDC Dashboard. I invite you to look at that on a regular basis, as we have numbers that come in, and also directions if you are wondering if somebody in your close surrounding is getting positive, what are the actions to take. So what are coronaviruses? How big are they? What are they good at? What are they sensitive to? So here is a little scale to just remind you that COVID-19 or SARS-CoV-2 is 100 nanometer diameter. It's a tiny little thing. It's about 10,000 times smaller than a period on your newspaper. It's about 1,000,000 times smaller than a tennis ball. So it's really, really small. So those spherical particles are encapsulating single-stranded RNA genomes. They are protected by a helical nucleocapsid, and they are enveloped into a lipid bilayer. So here we have this lipid bilayer that is sensitive to soap, to detergents. So that's why washing your hands will destroy the virus. And we have a few proteins, viral proteins, that are expressed on the surface. The envelope proteins, the matrix protein, and the most famous one, the spike proteins. And so those spikes are the focus of a lot of the vaccine designs. So I will go back and talk about the spikes here. But that's what gave the name to coronavirus, because it makes that crown on the coronaviruses. So coronaviruses, human coronaviruses are responsible for about 20 to 30% of the common colds. So we have alpha and beta coronaviruses. So just so you know, SARS, MERS, and the novel coronavirus are beta coronavirus, all of them. Okay, so we have the peak season for coronaviruses infection are between December and February. So we are at the end of the peak season for 2021. And we know there is seasonality in the infections. Infants and elderly can be infected. Infant will develop croup and bronchiolitis, while older people may develop pneumonia, acute respiratory distress syndrome, or even death in some rare cases. So coronavirus infect humans, but not only humans, they also infect animals. So we can infect pigs, cows, chickens, cats, dogs. So we know that cats can get COVID-19 actually. When you can imagine when you're a farmer and you have a herd of your porcine herd that gets contaminated, that's traumatic economic loss for those farmers. But it can cause severe disease in all these animals, okay? So viruses and viral entry. So how does a virus enter the cells and how does it replicate? So here is a human cell that is drawn here in that diagram, this is the inside of that cell. So SARS-CoV-2 will bind to a receptor expressed by that cell. So I want you to be familiar with that receptor. It's called the angiotensin converting enzyme, and it's the ACE2 receptor. I might call it like that in the future. So the ACE2 receptor is the receptor that is expressed on our cells that will bind that SARS-CoV-2. It binds to the spike. So that's why the spike is so important. And it binds via the receptor binding domain that is shown here in green. So once we have that binding, we have an internalization of that SARS-CoV-2 into the human cell. Once it happens, then the virus disintegrates and will replicate. So it will make more viruses. It will replicate its RNA to make more genetic material for the newer viruses, and it will translate the proteins. So now the M, the S, and the E proteins will be expressed in your own cells. And I love this drawing. I think it's very interesting to see the replication of the virus. But the only thing is, you have one virus that comes in, you don't have one virus that comes out. You have thousands of viruses that come out of one cell. That's where the amplification pattern occurs, okay? So here, this is a little bit of the work from Dr. Ralph Baric. So we know that coronaviruses, the natural reservoir for coronaviruses are bats, and we have millions and millions of bats around the world. They grow in bats and they mutate in bats. And that's where SARS-1, MERS, and SARS novel coronavirus have originated from. We know that they have a little mutation and they jump into different hosts. So now we suspect, highly suspect, that there was an intermediate mammal host, maybe a pangolin. That's what we are thinking for coronavirus, but it's still work in progress to know. And then there is another secondary mutation that will make it transmissible to humans. So that's where Dr. Baric, who in 2016 published a paper warning us about the threats of coronaviruses to human and animal health. He noticed that there are spontaneous mutations in that spike, and especially in the RBD, or the receptor binding domain, right here, or right here in that flat sequence here. And that can enhance cross-species transmission. So again, we want to pay attention to that receptor binding domain. And here I just noted for you the D614G, which is a mutation that started in Italy. And that mutation now is the most common strain in the US. So that started in Italy and took over in the US. But this is not the original mutation. You've heard about the UK variant. That's where it sits, right here. You've heard about the South African variant, and that's where in the RBD domain, that's where the South African variant sits. So all these mutations will change how the virus behaves. So either it enhances cross-species transmission, but it will also change, sometimes mutation outside of the spike can impact replication rates. So now we have viruses that are replicating faster. But all these mutations, either on the spike or other domains of the virus, can affect symptoms and disease severity. What we have seen, because we have the population that is getting closer and closer, animal population and human population, we have seen an acceleration of coronavirus cross-species movement in the 21st century. So again, we had SARS-1 in 2002, MERS in 2012, we had a porcine virus in 2018, and then now we have novel coronavirus in 2019. So it's really an acceleration of that. So what do we do in the lab to try to understand how SARS-CoV-2 behaves? Well, we grow airway human cells in cultures. So we have donors and we collect cells from those donors. You can see here, these are the lungs, right? And so we have a conducting airway that you can see in more details here. You have the airway epithelium, a lot of different cell types. It's too complicated for me to go into the details here, but this is what is gonna just line your airways. And then you have a little deeper, you have the submucosal glands that will produce the mucus. So when you're inhaling pathogens or dust, every day we do that, we secrete a little bit of mucus and it comes out of our lungs every day. Okay, and when we get sick, we have more mucus production. And so we feel it, we start feeling it. But even in normal conditions, we produce that mucus to clean our lungs. So what we do in the lab, and of course, you know, you have the conducting airways that go all the way deep into the alveoli, which are those little sacs that will allow the gas exchange and your breathing. So what we do in the lab is that we collect nasal cells for HNE, for human nasal epithelial cells, or cells from the large airways, large airway epithelial cells, or from the small airways, small airway epithelial cells. I just want to stress that because I'm going to talk about those three cell types later on. So what we do, we collect the cells and then we grow them on a petri dish, basically. We feed them with a growth media, we keep them happy for 4 to 5 weeks, and they differentiate into a pseudo-stratified epithelium. Okay, so they form really things that are very similar to our conducting airways in the lungs. So here is what they look like a little bit. So that's with the lid on, that's without the lid on. And you can see that little blob that you see on the center of all of these inserts is actually mucus produced by those cells. So they have cilia, they can transport the mucus around and they form those little hurricanes, we call them, because like North Carolina hurricanes, you know, they come in the center of those inserts and they transport the mucus. So the good thing about those models is that we can really use them to try to understand the physiology of different things, treat them with pharmacological agents, infect them, put them in contact with inflammatory cells, try to understand the physiology in the lungs. So here, I knew how to grow cells. I knew we had a coronavirus in Chapel Hill that had SARS-CoV-2. And then I said, can you infect some of my cells? And so he contacted some of his technicians, and that requires a BSL-3 facility, which is a biosafety level 3, which is one of the highest. You have BSL-4 for very, very rare and very contagious, but there are about 4 or 5 BSL-4 in the world only. So BSL-3 is very high protection facility. This is only to study biological agents that may cause serious harm to humans and maybe animals also. So you dress up for it, you infect your cells, you keep them in that, and you deactivate things afterwards. They never can come out infected. So everything I'm showing you is deactivated after for me to look at, but that's in the BSL-3. So Dr. Barrick does that for us, and actually Kate Edwards does that. And right after this talk, I'm gonna bring her some new cells, so let's not forget about it. So here, this is a little bit, so brace, this is a little complicated, but I'm gonna try to walk you through slowly. So you remember we have those nasal cells, large airway cells, and the small airway cells. And here, this is an apical view of those cultures, and in green is SARS-CoV-2 that we tagged with a fluorescent protein. So when you see a green cell, that's an infected cell. And you can see that maybe you have a dimmer signal in the small airway cells. Well, we could repeat that. We can measure how much viral proteins are replicated in those airway cultures. So here again, our nasal cells, large airway cells, small airway cells. And you can see that the virus in 0, 24, 48, 72. So within 4 days, really this virus takes over the cultures. So you can see, I want to emphasize how infectable nasal cells are. So that's why when you wear your mask, you want to cover your nose, not put the mask under the nose, okay? So that's super important because they are very good at replicating the virus. So here, this is just staining, and you see the apical view of those cells and you see the transversal view of those cells. In green, this is the SARS-CoV-2 virus, and in yellow, for instance, it's gonna be mucus cells, you can see here, and in blue, these are the nuclei of our human cells, right? So you can see how infectable those airway cells can be. So this is H&E here and large airways here. So I just want to scare you a little bit here to show you how nasal cells can be infected. So wear your mask on your nose, please. So this was published, by the way, in Cell in July by our group. So when I saw that and the infectiousness of this virus into our cultures, I couldn't help myself. I wanted to look closer. So for that, we use scanning electron microscopy, or SEM, because we can see the cell surface. It's like almost flying a drone on top of a forest. So imagine you're flying a drone and you see all these ciliated cells, so the cilia that are like moving the mucus out of the lungs, and you're looking for apples, right? The apples will be the SARS-CoV-2 virus. We are in a black and white world with electron microscopy. So I'm going to show you some of those images now. So this is the machine that allows us to do these very cool images at very, very high magnification power. So here are images of the lungs. Before I did any COVID research, I was able to do images on submucosal glands from the lungs. So this is an extracted lung, and we observed a gland that is actually spitting out the mucus. That is what is taking care of your pathogens every day in your lungs, right? So these images were taken by Cam Morrison, and he recolorized them with Photoshop from my lab. And here, this is again a mucus network. So this is the mucus here, a little more relaxed than when it just comes out of the glands, and it transports a red blood cell, several red blood cells. So it's pretty cool images, I think. So we knew how to do these images. We were interested in seeing the virus. So we took infected cultures under the SEM microscope. With Cam, we spent numerous hours looking at those until we found infected cells with those poisonous apples. And so here are the ciliated cells. So they have those cilia that are normally constantly beating and chasing the mucus out of your lungs. This is airway clearance. And you can see a little thread of mucus on top of those cells that is trying to take care of the virus, but it's only trying, because you can see that when we zoom into the roof of that cell that is here, you can see how many viruses are coming out of that cell. So that cell is spitting out tremendous amounts of viruses. So I hope I guided you to understand what we are looking at. This is an apical view of those ciliated cells that are spitting out virus through their membranes. So you can see how many viruses are between the cilia, and at high magnification power, you can see the spikes of the coronavirus. So Cam had fun, and he could recolorize those images, and he put the mucus in yellow, the cilia in pink, and the little viruses in blue. And so the cells are here in purple. So that's just to show you in images what the virus can look like. So those images were published in the New England Journal of Medicine. I think that was the first time somebody saw SARS-CoV-2 coming out of human cells or primary cells. They have seen them coming out of cell lines, but this is an airway primary cell. So that can tell us also what kind of cells are infected, which are the ciliated cells in your nose. And you need those to clear the mucus out, right? So one thing, after a long time, we let the virus incubate for a longer time. And then you can see that the ciliated cells are just not doing well. Once the virus comes in, it just gets hijacked. And they take over, they use those cells as factories of viruses. And you can see those cells are protruding out. They are bulging out. And trust me, but all these little dots that you see on that cell all these little dots here of SARS-CoV-2 viruses. So they swell, they start to detach, and they completely detach. They shed, they slough off the cell surface. And you can see that in infected cultures all the time. And that can occur within 48 to 72 hours. So that would be before people have symptoms. So we use TEM, that's another way of looking at high magnification power. Now we look at the belly, inside the belly of those cells. So this is a shed cell, and I'm using TEM, transmission electron microscopy. And you can see when I zoom in how many viruses this cell has. So those, again, do not forget that those transport in and out of your lungs. If they come out of your lungs, then you can easily infect the next person. If they go deep in your lungs, then that's when you can develop pneumonia, okay? So we have to be careful about those cells that are shedding a ton of viruses. So this is unpublished. We are planning to publish very soon, but I just wanted to show you a little bit the progression of these cell cultures. They are very nice. They have nice cilia. This is a cross section of our cultures in a Petri dish. After 48 hours, they start secreting maybe a little bit more mucus. They start having rounder cells. At 72 hours, you can see disorganization of the nuclei here. So really those cells are starting to struggle. At 96 hours, we have cells popping left and right. They are shedding off and they are transporting on top of our cultures. So again, we don't have the immune system here to try to cope with this infection, but just so you know, really in a few days, we have dramatic replication of the SARS-CoV-2 virus. So you can see here that apical view or aerial view of our cultures in SEM, and you can see them starting to protrude out and shed off very rapidly within 48 to 72 hours. So now we can correlate these images with virus titers. This is the infectious particles. We measure them, we can measure them. So you can see that they drastically go up between 24 and 72 hours, and then they kind of plateau. There is only so many cells we can infect in a Petri dish, right? In the lungs, it's different. And so we can also measure the mRNA of those viral proteins, and you can see again that it goes up and then it plateaus around 48 hours. And then we can measure also, I want you to just look at that black line at this point, okay? And then you can look at, we can count how many shed cells we have, and you can see that it's an exponential increase of the number of cells that are shedding. So of course, the idea that I want is to prevent that viral entry, prevent that viral replication. So we are trying pharmacological compounds. It's not published, so I'm not gonna say at this point what it is, but we have pharmacological compounds. We pre-treat the cells for 3 days before we infect them. And you can see in red here that we are trying to dampen the virus replication, and we are pretty successful at it. We have used different donor codes, different donor lungs, and we have seen the same effect every time. So this is in the pipeline, but it's unpublished, so hopefully it will be published very soon. So all of this to tell you, it's very important to wear your mask, but not every mask is equal. So if there is something to learn from this webinar, is what kind of masks work. And you can see here, we can see the particles that are secreted, and what you want is this number to be very high. You want to block the particle transmission, okay? So we know that N95 are very good, or KN95 shown here, are very good at blocking particle spreading. And then also the surgical masks are very good. What is not as good are the bandanas that I think are, I forgot where they are, but anyways, bandanas and gaiters, stay away from people that wear them because they are not effective. don't wear them yourself and stay away from people that wear them. The more layers, the better. The tighter the fit on your face, the better you are, okay? So as of this week, the FDA and the CDC has added one line that SARS-CoV-2 is not transmitted by food or packaging. So just as an FYI, you can relax to order some food from the restaurants, I guess. That's what it's saying. This is a very complex slide, so I feel a little bad for the non-scientist on the call, but I'm gonna try to explain to you how variants take over and how they dominate, right? So here we studied in the same models that I showed you with the airway cells, the nasal, the large airways, the small airways. We put in combination, we put the original SARS-CoV-2 before the Italian mutant took over with the Italian mutant. And you can see that in the nasal cells, the Italian mutant, which is D614G, which is an asparagine in the position 614 that is changed into a glycine. Details, details, details. That's not important for you. What is happening is that those variants are actually taking over. They are replicating much faster. It's significantly different, right? Whether we are looking at 24, 48, or 72 hours. In the large airways, maybe the replication is not as great, but you just know that the replication is in the nose is the starting point, okay? In the small airways, we haven't picked up any significant difference. What is interesting is that when we passage the cell, so now we are co-incubating the original virus with the variant, okay? And we are looking at the cells as we passage. So we are thinking we're passaging from one individual to the next, right? The next transmitted person. And so as we are doing those passages, we are looking at the genetic profile. So here, this is the original virus that gives you 2 bands on that gel. The mutant gives you only 1 band. So now we are mixing 50-50, that original virus with the variant, the Italian variant. And you can see as you passage more and more, then you go from maybe a mixture of both viruses to almost only the Italian variant. When you dilute more, when you put a lot more of the original virus compared to the Italian variant, so now 10-to-1 dilution, so you have 10 times more of the original, you can still see that it takes a little longer, but eventually that Italian variant takes over. So that's how we explain that the variants eventually take over. They are just better at replicating, at infecting, and they just, after a while, take over. So that was published in Science in November, okay? So we looked under the microscope at those variants. Are they different? Do they have more spikes? What's going on? And so you can see here, this is a TEM, this is SEM imaging. They really look the same. So I spent hours and hours counting on every single one of my images, the spikes. I could not see a difference. There was no significant difference in the number of spikes. So it's not because they have more spikes, it's probably because the spike might be a little more stable, and maybe there are other mutations that make them replicate much faster. So what we did is that we did a neutralization assay. So now we are presenting antibodies and the virus into the cells. So, and we are looking at which ones are stopping the virus and how fast they are stopping the virus. So we had people that were immunized that received the vaccine and they had their blood drawn, and we presented their antibodies to the original virus or the D614 gene variant, and we saw no significant difference in their way of neutralizing the virus. So for that specific mutation, the Italian mutation, there is no worries as far as the vaccination. However, we have learned recently that the South African variant might be of concern. The immunized people, vaccinated people might not be as prepared to fight this virus as the original viruses. So that's it for that side of the basic science. So now let's talk about the vaccines. because I know there are a lot of questions about that. And so there are a lot of vaccines. It's very confusing. mRNA, this is the first mRNA vaccine. So I know people are very, very confused about this. So just so you know, this is me getting my Pfizer vaccine. I have my 2 doses, so I should be immunized at least for the common variants at this point. And the goal of those vaccines is really to build up immunity. Our bodies have never seen that coronavirus. And this is why the virus is, and it replicates very quickly. So it's very easy for this virus to take over our body. If we have if we train our immune system to attack this virus, and this is what vaccines are doing, then we're gonna do a lot better if we are ever facing that virus. So we have in the pipeline, we have the Pfizer and the Moderna that have been approved, the AstraZeneca and the Johnson & Johnson. So you can see their efficacy rates, 95% for the mRNA and about 70% for AstraZeneca and Johnson & Johnson. So they've been approved. The first 2 ones are messenger RNA. So people are worried about that. How did we get this approved? And so just so you know, when there was the first SARS infection and the MERS infection early in the 2000s, we thought about those vaccines already. We had tested mRNA vaccines in animals then. So we have a little bit of setback to know the effect. You will not become a mutant. We are not internalizing the RNA into our genome. We express this protein for a few days. Our body sees it at the first time it sees it, it attacks it, builds immunity against it, and that's it. So the RNAs are only against the spike. So you cannot get sick. You will have symptoms, side effects, but that's vaccines. That's your immune system reacting. So this spike is the target of the vaccine. So we take just the RNA of the spike and we inject it, okay? So it expresses only the spike. No, the virus cannot be formed. And then the other AstraZeneca and Johnson & Johnson have the more common deactivated virus that we are familiar with, which actually our dear Louis Pasteur discovered. So just so you know, Louis Pasteur, he was studying viruses and he went on vacation and he left viruses in a jar. And he wanted to inoculate rabbits afterwards. And the rabbits didn't get sick. So that's for the little side story. And he could not understand what was happening. So those viruses got deactivated during his vacation. He inoculated the rabbits were just fine, right? It's not an active virus. And so he kept then he got new viruses. He inoculated the same rabbits, and those rabbits were not getting sick. And then he inoculated new rabbits that never saw the vaccine or the deactivated vaccine, and they were getting sick. So that's when the principle of vaccination occurred from our dear Louis Pasteur in end of 19th century. So, okay, so also we have a problem of storage. Now we know that Pfizer can be stored at -20, no longer at -80, which is a problem because not every site has -80s storage on site. So -20 for both Moderna and Pfizer, and they are producing a lot of them. I wouldn't, no matter what vaccine, we've been very vigilant at looking at the data, and they are very safe. So feel comfortable with that. So when will the US acquire herd immunity, which is when we are gonna all take our mask and live free again? Right now, we are about 15% of the population, I would say. As of tomorrow, it was predicted 10%. We are doing pretty good, but it's predicted that we're gonna get 50% immunity from the vaccine. Again, there are people that have had COVID-19 and they have some immunity. So who knows when we're gonna get that 70%, but the numbers will very quickly tell us when we are following the dashboard of Johns Hopkins, when we are in a safe zone. but you know, it's predicted to be in the fall of next year. Hopefully it's gonna occur a little earlier. Okay, so if you have fallen asleep, here are my conclusions. SARS-CoV-2 is responsible for the ongoing pandemic and will likely become endemic. So that means that it's here to stay. Sorry to tell you that. We are learning to live with it, and we are building immunity against this coronavirus, which is a great thing. That's what we want. We want to learn to teach our bodies to fight against it. The 3 W's are very effective. Wear, wait, and wash your hands. Wear a face mask, wait. If you're having to share an elevator with somebody, I always wait. I'm like, no, thank you. I'll see you later. And they are very effective at preventing SARS-CoV-2 transmission, but everyone needs to apply them. It's not because you're wearing a mask that you're protected if the person next to you doesn't, okay? SARS-CoV-2 infects the upper airway very efficiently, so it's very critical to properly wear your mask on your nose, please. SARS-CoV-2 infects also cells that release viruses before the onset of symptoms, and therefore asymptomatic individuals can transmit the disease. And we know that some people will stay asymptomatic anyway, even though they have the virus. If you have a chance, get vaccinated as soon as possible to prevent the spread of variants. So it's a race now, right? Since the variants are not neutralized as efficiently, we just want to vaccinate people before they can amplify into a large bulk of the population. So that is it for me. I want to thank everybody. And please, if you have questions, feel free to ask. And I think we have to use the chat if you have questions.
Yes,
or
yes. So maybe we can start the questions. I see maybe there's one first question. Oh, that's Jean-Marie Maillard. So hey, welcome, Jean-Marie. So Jean-Marie is maybe we can introduce him afterwards, but he says amazing photos. Thanks for explaining how they were made and presented. Great collaboration on the process. So Jean-Marie says, could you say, so Jean-Marie is in charge of epidemiology at the Department of Health in North Carolina, and maybe we can introduce him after the end of the questions. Maybe Jean-Marie, if you don't mind that would be introduce the two of you, that would be great. So his first question is, could you say a word about the early effects on cells and the marked effect on oxygen in blood? So a word about the early effect on cells and the marked effect on oxygen in blood.
So on cells, so those cells that are getting infected, we know that they are becoming apoptotic and maybe they have a secondary necropsis. So that's as far as I know in a Petri dish. So of course I do not have blood vessels in my Petri dishes. So I'm not gonna be able to comment on that. I know some people are working on patient's sputum and seeing some blood in it, but I will not go into that at this point.
All right.
Sorry, I cannot comment on
that. So Stephanie Gaub, I'm not sure how to pronounce it. Will the COVID vaccine be an annual vaccine like the flu vaccine?
It's possible. We are talking about boosters for the new variants. So once you have your full immunization, we are talking already about boosters to build the immunity for the South African variant, for instance. So it's a possibility. The good news is that mRNA is very rapidly produced and we can produce that in high numbers. And it's very easy to synthesize a small RNA. So if people stop being afraid of RNA vaccines, then that opens the door to really have very, very powerful vaccines in the future and rapid vaccines.
But the vaccine now the effect doesn't last very long though.
I think it does actually. The recent data are showing that it's a prolonged effect. Yes, very recent
when
data.
When you say prolonged,
the first people vaccinated were at the beginning of the trial. It cannot be more than 9 months anyway. So we don't know more than that.
Okay, all right.
But we know that people that were vaccinated, that were contaminated in China early in 2019, have been exposed and did not get contaminated. So even if it was a natural immunity, they have not. We know people have done the double contaminations, but it's rare cases still.
Okay, all right. From John Jacob, do the vaccines prevent spread or do they just prevent symptoms?
So the vaccines, a little bit of both, I think. So if you teach your immune system to attack the virus, you cannot replicate this virus just as much. Very recent data I heard from, Dr. Fauci in an interview saying that actually vaccinated people transmit much less or almost none this virus. So you cannot replicate the virus, you will be less contagious, more likely. And then, of course, same for your body. If you cannot replicate the virus, then you will not develop symptoms.
Okay. So from Antonella Longo, why are some people more susceptible than others to the virus? Are there any genetic differences on the receptor that could explain that? Can you try to grow cells on individuals with different receptors? So
yes, Antonella, my paper is coming out. So I know Antonella very well. So yes, we have done linear regression on the different codes of lungs that we have infected. So we have very highly infectable codes and not so infectable codes, and we don't really know what it is. It's not really related to age because the donors were in the same age range, but we see a correlation with the ACE2 receptor, that's for sure, and other things.
All right, and ACE2 receptor, could you remind us what?
So the ACE2 receptor is that receptor on our cells that is actually binding the spike and internalizing the virus, yes. So that the more you're going to have this receptor, the more likely you're going to have to trap viruses. If you have very few, it's just statistics at this point. It depends on the load of virus. If you have a high load, if you stay for 2 hours in an airplane with somebody that has COVID right next to you with no mask, that's a very high load. If you're outdoors, and you just cross paths with somebody that has COVID-19, you're not going to have a very high load. So all these statistics come into play, how much receptor you have and how much your load of virus is.
Do we know who has those receptors, or is it just random? We don't know who has more and who has less?
Yes, people with hypertension have higher rate of expression of those receptors, but we don't really know in the nose. So let's be careful here, because this is for the endothelial cells. In the nose, we don't really know what is the role of this receptor in the nose. We know we have a ton of ACE2 receptor in the GI tract, and yet only 5% of infected people will develop GI symptoms.
Interesting. Wow. So the more of the receptors you have, the more chances you have to be infected. All right. Wow. Interesting. Thank you. All right. Caroline Orense, I suppose that's a German name. What is the reason that deactivated vaccines are less effective than mRNA?
I would not know the answer to that. I just cannot comment on this, because deactivated vaccines is what we have used for a long time now. And so I just don't know what is happening on that side.
Okay. All right. That'll be something to follow up somewhere.
So I'm sure, yeah, the virologist and people are developing. Let me tell you that AstraZeneca and Johnson & Johnson are trying to understand why it's not 95%.
I'm sure.
I'm pretty sure. I'm not going to go into that race.
And other people too, other companies too, I'm sure. So how do RNA vaccines work? Is RNA, okay, English, does RNA remain or can it be replicated somehow? Does the RNA penetrate the cells?
Okay, so the RNA has to go inside your cells. So the RNAs are injected with lipids just to make it into your cells because it needs the machinery to create a protein, right? The immune system will not recognize the RNA per se. It will recognize the protein as a new protein, right? So it needs to go into your body. It needs to go into your cells. It needs to replicate exactly like I showed in the little diagram. It needs to replicate and form those proteins, but it's just floating in your cell and it will extinguish within days. So we don't keep producing or translating this mRNA forever. It doesn't go into your genome. We don't become mutants. It's not inserted in your genome. It's just floating in the cytoplasm. It gets translated and then it dies like every other RNA. RNAs don't last forever. The genetic, like DNA lasts forever. The DNA in your cells will last forever and will be transmitted from one cell to the next. But RNA just have a short lifespan. And that's what is happening with the RNA. It gets translated into protein. That's very important, right? Because the immune system will not recognize the RNA. It will recognize the protein.
Right. Well, that's
so we are making that, we are using our own cells to be the factory. So before, what we were doing is using cells in Petri dish to produce protein of viruses to create the vaccines. Here, we are deciding to use our own cells as that factory. So it's already sterile. It's already, you know, we have all the amino acids to form this protein. And so that's why this was a great idea. And I think the lady that thought of this idea, she deserves the Nobel Prize.
She probably will have the Nobel Prize, yeah. I'm sure.
Yes, yes. Great idea.
Great, yes. So Lakeisha Shaw, how does the side effect of the vaccines, have an impact on patients that are dealing with other medical issues? That's a good question.
Yeah, that's a good question because we know that, so I'm not gonna comment on immunosuppressed people because they are a very totally different category, but you wanna make sure that when you're getting your vaccines, you wanna be in very good shape, in good health. You wanna drink a lot, you wanna take vitamins weeks before when you sign up for your vaccine. So take your vitamins, be as good of a shape as you can be. I cannot comment on very particular cases, depending on some health states, but we should be strong enough to handle that, the strength of those vaccines. It's just stimulating our immune response. So what I've heard is like maybe older people don't have a strong immune response, but maybe younger 20-something will have stronger side effects.
Oh, really? So have you heard of about any cases or any diseases or comorbidities that people, that the researchers have said those people can't have the vaccine, it would be too dangerous for them? Or
immunosuppressed, I think we have to watch people with allergies, I know of. After that, I would not comment on people that are very critically ill. I don't know if they decide to do it or not.
Yeah. All right, and allergies, yeah, you never know. Before you have an allergy, you never know what's going to happen. So from Mosde Bagheri, how does the RNA vaccine work? I think we've already said that. Does the RNA remain, or can it be replicated somehow? The RNA, does it penetrate in the cells?
I know everybody is a little worried about that. I think we need to do a little video on the web about that, and explain that it doesn't enter your genome, and therefore it's not there to stay.
Yeah, that's great. That's, if there's one sentence to remember, that's probably okay. From Jean-Marie Maillard, herd immunity will also need to have most persons who are offered the vaccine to accept them. Hopefully more will, with confidence building up with time. Jean-Marie, you need to say a word about that, more words about that. That's very good. That's very good.
I do agree. I have to say that really I had some skeptics. At UNC, we were so lucky to have the vaccine very early on. people were skeptical at the beginning, and they see their neighbors, they see their colleagues getting the vaccine, and oh, you just had a swollen arm? Oh, I'll do it. Why not? And so people get a little more confident. I can tell you, my first shot, just a sore arm, nothing else. Second shot, I did not expect to do anything the next day. I was like, I might be out of work. No, I was fine. I woke up, I was like, just a little sore. I got my coffee. I was like, oh, I'm actually doing good. The only thing is my lymph node on my arm, the ones that was injected, were swollen and painful. So I know I had an immune reaction, which is what I wanted. I was like, great, that's great. I'm building antibodies here. So
interesting.
This is why I thought I would put a picture of me getting the vaccine, just to show you I did it.
Yeah, I think you're right. You're correct. As the more people do it, the more people will do it, because they'll see a lot of people.
My husband did it. You know, I know so many people that have been vaccinated now, because I know a lot of people in the healthcare domain, and they are doing very good. Kendall, Kenza, Cameron, the people in my lab, because we work on the virus, they've been vaccinated.
So from Lakeisha, will this vaccine need to be taken yearly? Like, oh, we've already asked that. Like the flu shot, will it just be the one-time vaccination that protect you for a long period of time? So we've already answered.
I think we might need boosters for variants. I don't know. We'll see how the immune system works, but.
From Michelle Washington, will a recording be available so I can watch it again? Yes, it will be available. You can go on our website, the French American Chamber of Commerce website, and it's facccarolinas.org or com. Can't remember. And you can rewatch the program. I mean the webinar. Antonella Longo. There is a lot of opposition to vaccines in France. I am surprised by that. Can you comment?
Are you surprised? French people against something new? No, I'm not surprised. I just think that, just like we mentioned, they will relax as they see more and more people getting vaccinated. My mom got the vaccine. I'm very happy about that. I was very worried about her, so I'm super happy. And people like her saying to all her friends, I got the vaccine, and she's finding a little bit of resistance. Really? You got the vaccine? How did you do? And so I'm going to call her, because she had her second shot yesterday. So I'm just going to call her after that and see how she's doing. But she did great on the first one. She barely felt it.
I hope there is a little bit of shaming, you know, because we see Israel and
yes, 78%.
You know, the United Kingdom are doing really well, and we are just lagging behind. It's really a little shameful.
I know. They should inject all the arms that they can. People that are not skeptics, they should just inject them, and that those people become, you know, a testament that it's actually working and it's not that bad.
I think there's also a problem of approvisionnement. I mean, it seems like there are not enough vaccines for some reason, but that's a problem for, you know, for the politicians, I guess.
Yes, so, exactly. I'm not going to go into that.
I know, exactly. So Caroline Orenze, does it make sense for people who tested positive to get the vaccine at this point? This is a great question.
Yes. So I think in France, it's only one shot for people that have tested positive. But I would say, yes, get vaccinated. Because what if you were asymptomatic and your immune system was really not stimulated? Then you're going to get in the room thinking you're immune with somebody that has a boatload of SARS-CoV-2, and you're going to get sick. So please, the vaccine, yes, I would. You might not be a priority though, because you might have already some immune reaction, but I would not be worried to boost your immune system to see it again. You're going to really be prepared for that.
And can I say, I mean, I have because you read all the stories on the internet and you never sure if they're real or not, but I have a cousin in France who actually got it in April, very lightly, and he re-got it in January, and he's been in the hospital for 3 weeks, and he almost lost his life.
Do you see
so
that?
You can get it again, and it's worse.
That's where the load is playing a role. How heavy you get hit the first time, or is it just one day your nose, or you get it into your lungs, and now that gets really accelerated very quickly.
I remember you telling me you have to blow your nose a lot.
Yes, don't hesitate, get your Kleenex.
Okay, Jean-Marie Maillard, good point mentioning if you experience some side effects, that's evidence your immune system is reacting. As you said, that's when it is wanted. Excellent.
Yes.
From Nicole Schwerbrock, good job, Camille, NFSCC. From Christophe Henrion, to amend your comment, my mom got COVID-19 in November and got both doses of the vaccine. So she is commenting on that. From Florence Henon, how come some people react so much more than others to the second shot of the vaccine and why?
I think it's about the immune system. I think I'm in good shape, but, I did fairly good, but I'm also, probably used to having like some pain. And so that wasn't a super big discomfort for me. But I know people have headaches. I have headaches all the time. Maybe I had a headache. I didn't even feel it because for me, it's not that traumatic because I have headaches quite often. I just don't know. I really think it's because of the strength of your immune system.
Yeah. There's one last question here. Is there an explanation about the children's immune system versus in front of COVID?
Yeah.
Yeah.
I think this is a big player why they are not getting infected very easily.
Is it?
I believe in that. Yeah, they have a super strong immune system and they are young. It responds very quickly. So it's a fine balance between how the virus is replicating and your immune system like taking it down, because it can go out of whack very quickly and your immune system cannot catch up.
Yeah. Now I have a question a little bit with that, because a question that comes up often is, if someone gets the COVID, how long can they transmit it to you? I mean, can they transmit it even after they have no symptoms? I mean,
yes. You want a negative test, and just stay as long as
they have a negative test.
2 weeks, I think, from the start, from the exposure day to, but you never know really where you are in the infectious scope.
Exactly.
Are you picking it up right before? So I would say, stay clear and wait 10 days. If you have been tested positive, stay 10 days without.
Yeah, just a lot of people are worried about how infectious a person is, how long they are infectious afterwards. Okay, so probably even 3 weeks, maybe even better. Yeah,
but then it's hard to stay in your bedroom for 3 weeks.
No, I'm a director of a school. I'm just wondering if someone just okay. I have a question about the cross-contamination. You said that animals can transmit it to humans. Can humans transmit it to animals?
Mm-hmm. Yes, absolutely.
And how about pets?
Yes. This is why actually when you have a household that has COVID-19, like you have 2-3 people in the same household, if they have cats, do not let your cats go out because they can carry it in their feces. Wow. So they will have GI symptoms, cats, most likely. But we know we can infect ferrets and cats with SARS-CoV-2. Wow.
That's incredible. Wow. I mean, it's amazing. Yeah, I think, can I ask you a question, maybe, what your research is about? Is it about a medication?
It's more trying to understand how the cell react and how we can just stop the progression of SARS-CoV-2. I am not trying to make money at all. This is not my forte. It's just really trying to figure out a way that I can slow down the progression of SARS-CoV-2 to help the immune system take over before the virus just multiplies. I'm thinking about a nasal spray, nothing more, no injection, just a nasal spray.
But that's something you would take as a preventive measure.
Mm-hmm. All
right. Wow. Something you put in your, oh, wow. Okay. That's very interesting. That's exciting. Camille, you have to come back on the webinar. So there's another question. Is there really a relationship between blood group and COVID immunity? in particular for O group, because I've read that over the internet.
Yes, I know. O groups are a little more protected, and so from what I understood,
Really?
Yeah, I don't know, right? I think this is, I just don't pay too much attention on this kind of stuff because I think there are people in the hospital, in the ICU bed from all the groups. Otherwise, we would have figured it out by now if one group was very protected.
Okay, that was from Ludovic Casas.
But I know there are discussions about that, yes.
Yeah, interesting. Wow. Can I maybe I don't know if Jean-Marie would like to be seen, but I mean we would like to talk to you, Jean-Marie. I mean, would you let's see. I will promote to panelists.
I want to thank everybody for attending and asking all these fantastic questions. First of all,
Jean-Marie, here, Jean-Marie. Jean-Marie, are you available to introduce yourself to Camille and to the rest of us?
Je suis désolé, merci parce que je pouvais pas voir comment parler ou.
Oui, no, I have to promote you actually.
I understand.
So. All right. Hey, so could you?
Yeah, it's great presentation. Thank you very much. I think you explained a lot of the principles that people may have become, or I've heard many times over the past year. But it's great to have a specialist showing exactly what happens. And to me, it was like when you had these pictures of how many hours after you start the culture, you see the effect showing, it immediately made me think, that was my question, about people who don't realize that their oxygen level falls down dramatically. And sometimes that's why they don't go to an emergency department. So it was great to see your physical evidence of what goes on, and that might be behind the.
Yeah, that cell shedding cannot be good for gas exchange. That's for sure. Yeah. And plus, we might have, broncho obstruction in very deep lungs. So we know that the alveolar cells can be infected by SARS-CoV-2. So if you inhale the virus really deep in the vacuoles, in the alveoli, then that's where the gas exchange can be compromised.
Yeah, and great also to see the differences between the cells in the nasal walls. And as you go down, the difference in susceptibility in some ways, but so protecting first level of barrier protection to protect the nasal mucosa is a great point. Yeah, thank you.
People, as you say, Camille, people put that mask on your nose.
Very,
the more time we have, I mean, as time goes by, there's more and more evidence of how protective they are.
Yeah.
And they are actually pretty, so thank you also for mentioning the 3 Ws. And it's critical to continue, because when you hear about the threat of the new variants, there are concerns also that, it's better to continue to use the control measures that we have. They work well. The problem is, for a long time, there are lots of people who were not applying them. And so we're in better shape. I think between that and the vaccine and the good news we hear about boosted production of the vaccine, we have reasons to be optimistic. There are a few things just for precision. The North Carolina data are slightly different from the ones you had.
Jean-Marie, could you introduce yourself, because I'm not sure people know.
Oh, sorry. So I work with the North Carolina Department of Health and Human Services, and like Camille, I came many years ago, and it's been actually, in my case, a fantastic professional experience, because we went through very significant events like anthrax and SARS and pandemic influenza H1N1 in 2009, and now COVID. And each time we, just quickly, the public health system usually is not getting much at all. These events made people realize of how important certain things that public health professionals do is, and therefore we got resources, and we use the money that's directed to state public health systems each time to try to build up infrastructure rather than to limit it to the response to the agent of the day. Anyway, thank you. This was a fantastic presentation.
Yeah, I'm sorry if I don't have the same data as you.
Oh, I just wanted to say, positivity rate is 6% now. So
Okay,
it's a
so
little
it's
lower than the
dropping. Okay,
8%
I think they
showed.
don't refresh.
Yeah,
I know it's
they
dropping,
don't refresh.
and I thought we were around 5. And then when I went yesterday onto the Johns Hopkins website, I'm like 8. I'm surprised. I double checked, so I know these were the numbers from yesterday, but I think they just don't upload every day.
Yeah,
So
every time you see a different source,
let's do
you
it.
have
We
slight
can do it.
differences. But the good thing
That's
is all the
great.
trends, all the direction of the trends is in
Yes.
the good direction.
Yes.
So that is a great
Keep
thing
wearing
to
your
see.
masks, guys. I know it's a bit of a problem. It's kind of annoying, but it's such a nothing deal. If that can save life, it's nothing. It's really nothing.
And as you said, I think maybe your better argument was for the mask and the vaccine was to prevent the new variants.
Yeah, we are in a race with them. So
that's great. That's great.
We just need to prevent them from growing into the population and mutating again and becoming even more virulent. So that's we are in a race.
Yeah,
we're gonna win.
We are.
I know it.
Thank you. Well, thank you very much, Camille. This was an excellent, wonderful presentation. We really enjoyed it. And everyone, it will be available on our website, FACC Carolinas. If you want to get the newsletter, tell us, and we will send you the newsletter. You'll get it also in our newsletter. And thank you very much, Jean-Marie, for intervening at the end. And
nice meeting
everyone. And thank you for both of you for your work, because to prevent the virus from spreading in North Carolina. Thank you very much. Thank you.
Bye. Thank you, everyone.
Bye.
Bye-bye.